Excellent. Great. Thank you, Jonathan. Thanks very much, everyone, for joining. It's a pleasure to welcome to our current ECSA Files seminar in March, Iliana Baums. Iliana got a PhD from the University of Miami. She then went on to do a postdoc there as well with Dr. Jack Fell. From there, she moved to University of Hawaii, sounds nice, as an assistant researcher. And now she has gone back. Oh no, then she spent time as a professor of biology at Penn State from August 2006. And she now recently, I think 1.5, 2 years ago, came back to Germany, and she now leads the marine conservation group in the Helmholtz Institute for Functional Marine Biodiversity at Oldenburg University. So Iliana, it's really great to have you. Thanks very much for joining us. And I'm very much looking forward to find out more about how corals adapt to rising temperatures.
Well, thank you so much, Stephanie and Jonathan, for inviting me to talk to you about coral adaptation. I, as you just mentioned, moved back to Germany after 20 years or so in the US and lead a research group at the new Helmholtz Institute for Functional Marine Biodiversity at the University of Oldenburg. And the mission of this institute is to provide a scientific basis for marine nature conservation and ecosystem management. And so in keeping with this mission, my aim today is to unravel our current understanding of how corals may adapt to rising temperatures as the basis for coral conservation and restoration interventions. So ocean warming impacts corals, and the narrow thermal tolerance of corals and their symbiotic partners raises the question about their adaptive capacity. We think that the evolution of de novo host adaptations is thought to be too slow to keep pace with this climate change. And that assumption underlies some of the models that have predicted the demise of reefs in the near future. However, there's evidence that selection can act on existing variation that is already there, and so increase the frequency of stress-resistant alleles. And this process is more rapid than having to wait for de novo mutations to arise. We can use these insights to develop concrete advice for restoration activities. Corals are sensitive to even slight increases in ocean temperature, and that is because corals live in close association with single-celled algae in the family Symbiodiniaceae. These algae in the Symbiodiniaceae present a large diversity of genera, species, and strains. In some cases, the association with host and symbiont species is both specific and stable. But when temperature rises just 2 to 3 degrees Celsius above normal, dysbiosis occurs and the algae are expelled from the host tissue. And this process is called coral bleaching. The exact cause of coral bleaching is not well understood. Nevertheless, the consequences can be dire. So you can go from vibrant, diverse reefs here in an image from Charlie Veron from the Great Barrier Reef to completely bleached white reefs. If the temperatures don't decrease in time, these bleached coral colonies may die and get eroded over time so that you can see these fields of rubble. Corals have bleached, as long as we know, and they're adapted to these large-scale disturbances with an amazing ability to re-emerge from these fields of rubble, both by regrowth of patches of tissue that remain behind in the rubble, as well as through the arrival of larval recruits that settle onto the benthos and grow into new colonies. However, the ability to recover from large-scale disturbance events depends, one, on the availability of larval recruits, and two, on the frequency and severity of these stress events. Unfortunately, we've seen both an increase in the severity of the stress events. So the maximal temperatures that are reached each year are becoming higher and higher, and the frequency is higher. So the time between disturbances is so short that coral recovery is imperiled. This past summer in 2023, we had one of the worst high temperature events on record. These here are data from NOAA that show record high sea surface temperatures in the Florida Keys that were much higher than the average for the past since 1985 or so, since we have measurements. And these high temperatures lasted for so long that the cumulative stress of this heat stress resulted in widespread bleaching throughout the reefs in the Caribbean and also in high coral mortality. Given this increasing change in environmental conditions, we want to understand whether there's a capacity of these coral populations to adapt to these rapid changes. In order for me to discuss this, I will give a brief sort of summary of how we think selection and adaptation may work. This is to, get everybody in the boat, no matter what your background. So I will begin by summarizing some basic tenets about how adaptation is thought to work, and then I'll apply those tenets to corals in particular. So the first is that selection enables adaptation. So selection is a process in which environmental and genetic factors together determine if an individual thrives. In this example, you have a recent change in the environment that resulted either in large trees or a herd of giraffes has moved into a new area with particularly large and tall trees. And in this case, the giraffes with the long necks eat and survive, and the ones with the short necks will starve. The second tenet is that selection acts on the phenotype of an individual. The phenotype stems from the genotype, so the genes that an individual has, and its interaction with the environment. This is really important. So in this example, the selected phenotype is the long neck in an area with tall trees. The long neck wouldn't be useful in an area with short trees. The third tenet is that adaptation can happen to a population when an individual with a selected phenotype, so for example, being tall, passes that phenotype on to its offspring. So it's important that the trait is heritable and that the population reproduces. Finally, we can detect the effects of selection by looking at shifts in allele frequencies in the population. So this is because individuals that have an adaptive allele have higher reproductive success than those that do not carry the allele. This is the basis for our understanding of Darwinian selection and adaptation. Okay. So now what we're going to do is we're going to use these basic tenets and apply these to corals in particular to discuss how corals adapt and how we may use this information to help in coral restoration. Finally, if I have time, I will also talk a bit about the role the coral symbiotic algae play in the adaptation of the holobiont. So that's the coral and the algae together. So we'll start here with the first, selection enables adaptation. We can study this in corals by generating controlled crosses. So this is, crossing colonies of one genotype with eggs and sperm from another. And then we can expose these larval cultures to increased heat stress. We've done this both in Curacao and Puerto Rico, and just assay the allele frequencies in these pools of larvae. And look if there's a shift under high temperature. And indeed, we see such a thing. There are significant parallel shifts in allele frequencies between temperatures at day 1 and day 4, observed in all samples. And this is consistent with selection on standing genetic diversity. So basically, some of the parents that contributed to these larval cultures had alleles that helped their offspring in particular deal with the higher temperature better And those are the ones that survive, and the ones that had alleles that are not so good at dealing with the higher temperatures died. And so their alleles were removed from these larval cultures. Now, the second tenet is how selection acts on the phenotype of an individual. In reef-building corals, these are colonial organisms. So individual colony is a really confusing term. So corals are often built these big thickets here. You can see these images are from Belize. The top one is the Alcon coral, Acropora palmata, and the bottom one is a sister species, Acropora silvercornis, in Belize. And both species form these large thickets where it's very hard to see where one colony starts and another one ends. And you certainly don't know which one is an individual or a new genotype compared to another. So to explain this a little further, I have here a cartoon of the life cycle of these corals. The corals we're talking about today are hermaphrodites. So they are produced sexually by releasing large amounts of gametes during an annual broadcast spawning events. The eggs are typically fertilized externally by non-self sperm. So they need sperm from a different genotype in order for fertilization to succeed. This results in new genets, this is how we call them, because the parental genomes get reshuffled during meiosis and recombination. Then after development, these pelagic larvae eventually settle onto the benthos, and there they metamorphose into a primary polyp. So they go from a planula larvae into this primary polyp. And then through asexual reproduction, so this is, for example, through budding, and also through colony fragmentation, an individual Palmata juvenile can grow into these multiple large coral colonies. We refer to each of the physically separate colonies as ramets, and they all belong to the same original genotype or genet when you can trace that tissue back to this meiotic, through this fertilization event between a particular sperm and an egg. So while individual polyps and entire ramets or colonies can die for a variety of reasons, the genet itself can survive for centuries and grow to cover hundreds of square meters. And that way they're very similar to seagrasses that also have this ability to asexually reproduce via their rhizomes. And part of the seagrass bed may die back, but the genet itself can persist for a very long time. So similar to how the terminology that is used for seagrasses, we use the terms genet, ramet, and genotype. So a genet is all of the tissue grown from one primary polyp. All that tissue belongs to the same genet. Sometimes people also use the word clone. And a ramet, those are physically independent modules arising from colony fragmentation or other asexual means. So a genet can have one or many member ramets or clone mates. The reason why I don't usually use the term genotype is because the genotype is the genetic makeup of a sample for a set of genetic markers. This could be SNPs or, say, microsatellites. When you have enough markers assayed, then a sample can be assigned to a genet based on its genotype. Now, asexual reproduction is common across the cnidaria, not just the corals. This includes also the jellies and, for example, fire corals. Here, this is an example for a fire coral. This was work by Dubé et al., and they sampled fire corals along a transect, 300-meter-long transect, going from onshore to offshore. And all of these dots are colored for their genet identity. And you can see that some of these genets extend over hundreds or so meters from the back reef to the reef slope. In the Caribbean, we have a very similar situation. This here is an example of the Alcon coral, Acropora palmata, where we sampled them using a standard randomized sampling design, and we could show that some reefs are dominated by just one genet. So in the upper polar plot, you can see here, all of these dots are colonies that have the same shape, they're the same genet, and they also host the same symbiont strain. That's the fill, it's all blue. In Curacao, we have a bit more diversity. We have several different host genets indicated by the different shapes here, as well as a few different symbiont strains that are found in those host colonies. We have known for many years that corals reproduce extensively via fragmentation. In fact, some of the common statistics used to describe genotypic diversity in plants and animals were based on coral studies by Stoddard in 1983. So this excursion was important because the second tenet says that selection acts on the phenotype of an individual. In order to define individual for a coral, we can now say that we should use coral genet as the unit of selection, which fits most assumptions of evolutionary genetic models. The second question then is, how do we detect genets? With the advent of next-generation sequencing, we can now assay genetic markers throughout the genome, and this gives us the power to distinguish between closely related individuals, such as full siblings, indicated here in this bar graph by the hatched bars. So here, these are the full siblings, and we can distinguish them from ramets of the same genet that are nearly identical right here in a plot of these pairwise genetic distances. You may notice from the white bars that in this particular species, again, this is the elkhorn corals, most individuals have intermediate pairwise genetic distances between them. So these corals are generally not inbred and instead show a normal distribution of these pairwise genetic distances. So now that we figured out what an individual is, let's turn to the second part of this tenet, which is the phenotype. So selection acts on the phenotype of a genet. We know from many field observations as well as from field studies that there is phenotypic variation in the holobiont with respect to stress tolerance. And this is quite obvious from this image here. These are two colonies of the same species. This is Porites asteroides. There are one here is fully pigmented and its neighbor is completely white, even though they clearly are exposed to identical environmental conditions. They grow right next to each other, and yet one is bleached and the other one isn't. And we know that genetic diversity of the host contributes to this holobiont stress tolerance. We thus want to measure these phenotypes in a standardized way, if at all possible, so that we can start to understand what is the distribution of these differences in phenotypes within species. And this has recently become possible by this development of a sea bass system led by the Barsh's lab. And what they've done here is they've basically miniaturized coral aquaria in such a way that you can stick them into suitcases and travel with them wherever you need to go to measure these phenotypes in coral populations. This here is an example of our sea bass system that we set up in the Virgin Islands. And it consists of these 4 little coolers that each can be controlled very nicely in terms of their temperature and as well as the light. And that allows for very standardized experimental setup so that we can expose coral colonies from across their ranges to a standard set of environmental conditions, of stress conditions in the lab. And the kind of short-term heat stress assay that we've developed here includes a short ramping period and then a temperature stress treatment that's very high temperature stress that we expose these corals to. But the exposure is very short. It's just a few hours before the temperature returns to the mean monthly max, and then they are allowed to recover overnight. And throughout this short-term heat stress, we assay them for various parameters, including gene expression and the chlorophyll sort of photosynthetic performance of their symbionts. Now, short-term heat stress experiments have strengths and weaknesses. The weakness of the approach is that it's unclear how the short-term heat stress tolerance in the tank, as measured through this PAM fluorometry, translates to survival of corals in the fields after they've been exposed to many weeks of heat stress. This relationship can be estimated, of course, but whatever the outcome, I think it's a true strength of the approach that we can have a very standardized experiment across species and oceans, and that allows us to benchmark the coral heat stress response against which we can then evaluate other phenotypes. And this is exactly what we're doing at the moment. We have a big collaboration that aims to benchmark these stress phenotypes globally. So there's groups in Australia, in the Central Pacific, in the Red Sea, in the Arabian Gulf, as well as in the Caribbean, where we're working together to measure these stress phenotypes. So this is a collaboration between the Volstar Barshes, Bay, Venezuela, and Baums Labs. It's funded by the Paul Allen Foundation, NOAA, and other funders. So some of our early results were really interesting with respect to how they relate to past heat stress events. So we measured the heat stress response of corals in Florida. This is a map of the Caribbean, in Florida, here in the Virgin Islands, and in the Southern Caribbean in Curacao. And what's plotted on these graphs as actually the spatial variability of heat stress exposure in the Caribbean, derived from satellite imagery. And we measure this in degree heating weeks. It's basically accumulation of heat stress in sort of rolling windows. And here on the right, what Kelly Gomez and my group plotted is the number of these heat stress events measured in degree heating weeks. That's the term we use for this, in Florida, in the Virgin Islands, and in Curacao. You can see very nicely how these regions in the Caribbean have had, in fact, differences in how they experienced heat stress in the past. And we were wondering whether or not this past heat stress experience influences the stress response of the corals, which would be indicative of ongoing adaptation to these heat exposures. So here, this is the type of data you get from the short-term heat stress exposure. What is plotted here is the standardized heat stress applied to 4 coral species from 2 reefs in St. Thomas. Each population was represented by 12 to 14 genets, and we measured their photochemical efficiency over these treatment temperatures. So the treatment temperatures are here on the bottom, and then on the y-axis is the density plot of the photochemical efficiency. So the black lines show the temperature at which the holobions of that population lost on average 50% of their photochemical efficiency. And this is like a typical LD50 type measurement. So it's notable how the species differed not only in the mean value of these ED50, but also in the phenotype space they occupy. So Porites asteroides down here occupies a much wider space, say, than Acropora palmata, the Alcon corals, which we find much narrower responses. So from the same pulse amplitude modulation data, we can not only calculate the photochemical efficiency, but also the damage that heat inflicts on photosystem 2 of the symbionts. And so here, Kelly plotted the accumulated damage of photosystem 2 against the controls. So in Curacao, which has experienced much higher heat stress events above 8-degree heating weeks in the past, the corals didn't show much difference between control and heat stress fragments. Whereas in the Florida Keys, the heat stress fragments accumulated a lot of damage compared to controls. And not only did the damage accumulate was significant in the Florida Keys compared to Curacao. The repair of this photosystem after heat stress had stopped during the recovery period was also much faster in Curacao. So overall, the Curacao corals resisted the heat stress very well. And so this is consistent with a adaptation to these past heat stress events that has worked well or has already taken hold in Curacao, but perhaps not yet in Florida. So the third tenet is that adaptation can happen to a population when an individual with a selected phenotype passes that phenotype on to its offspring. So it's therefore necessary that the phenotype is heritable and that reproduction occurs. Now, the evidence for this in corals is fairly young in the sense that we've only been able to look at the first generation of corals that were assayed for this particular purpose. This here is an example from the Persian Gulf and the Indian Ocean. So we know that there's a genetic basis for thermal tolerance in coral hosts, and that this thermal tolerance is heritable. And several papers have shown that breeding for heat tolerance is effective, at least in this first generation. Let me see my front here. Here are some of these papers. For example, Howells et al. Crossed colonies from the Persian Gulf that live in very hot waters with colonies from the Indian Ocean that live in relatively colder waters. They also produced hybrid families between these two regions. Now, there was no difference in the temperature tolerance in the larval families, the cold temperatures, but at the hot temperatures, the Gulf and hybrid families had higher survival. So this here is percent survival. The red is the Persian families. These are hybrids, and this is the Indian Ocean families. So at 36 degrees, the hybrids and the Persian Gulf families had higher survival compared to the Indian Ocean families. So this is an indication that by selecting hot water adapted parents, we can produce offspring that has higher survival in hot water. What is unclear is how much of this enhanced survival is due to selection at the DNA level for different alleles, and what portion is contributed by the parents or maternal effects. To entangle this, we actually need F2 or later generation larvae, and that is work in progress, but not yet published. And part of the reason is that coral life cycle is quite long, so it takes 3 to 4 years for larvae to become adults that are reproductive. So looking at these phenotypes here, these are plotted now by temperature across the different species. And it's tempting to select just the holobionts for breeding efforts that have the highest LD50 as parents for the next generation, and so hope to increase the thermal tolerance of the species. And strong artificial selection can lead to rapid adaptation, and this is also called genetic gain in the animal breeding literature, but it does often result in a few individuals contributing to the next generation as parents. And we would then have low effective population sizes. So if we have only a small number of founders together with these random changes in allele frequencies that occur from one generation to the next, we can have a loss of genetic diversity. So it's therefore difficult to balance these goals of rapid adaptation to changing temperatures with maintaining high genetic diversity. And this can be achieved if one makes genetic management plans to balance the need for rapid adaptation and maintaining a high genetic diversity that may buffer against future challenges such as novel diseases or novel competitors. So the final tenet here is that selection causes shifts in allele frequency in the population. And I showed you an example earlier how we can study selection experimentally using these coral larval cultures exposed to differential temperatures. But you can also look for the long-term effects of selection in natural coral populations. And here at Rowley Shores in Western Australia, this is a study that looked at the prominent reef flat that comes exposed at low tide, and there's stagnant water in these lagoons here that heats up, and so they present as natural laboratory for characterizing a mechanism of coral resilience. So in this case, the authors collected Acropora tenuis from the lagoons and the fore reef, and then looked at both the algal symbionts as well as host gene expression and the genetic composition of these populations. And they could show that there were certain loci associated with the ability to survive higher variability and higher temperature stress. And there was nice shifts in the allele frequencies at those loci between the inner lagoon and the slope, which were physically, in fact, very close to each other. So this result highlights the potential for spatially varying selection across complex reefscapes that can, really drive ecological divergences in these climate-related traits over small distances. In fact, this evidence for local adaptation is widespread. We have inshore-offshore gradients, there's depth gradients, and there's latitudinal gradients in corals that we can exploit to find differences in allele frequencies and local adaptation. So I think selection is ongoing in coral populations, and genet and habitat diversity fuels this adaptation. Now, what does this mean for restoring coral populations? We can basically apply the tenets of selection to coral restoration and conservation to come up with a plan that helps us maximize the adaptive potential of restored populations. So to enable the persistence of coral populations exposed to severe selection pressure from the many stressors we have in today's reefs, not just temperature, but also sinking pH and increased eutrophication, we recommend a mixed provenance strategy. So we should source genets, both locally as well as from more distant, environmentally distinct sites. We estimate that sourcing just 3 to 4 genets per reef along environmental gradients can capture a majority of interspecific genetic diversity in these species. In addition, assisted gene flow can help increase local allele diversity. So practitioners should then propagate genets with one or more phenotypic traits that are predicted to be valuable in the future, such as high growth rates or increased heat stress. And in addition, to encourage sexual reproduction, which is important to enable adaptation processes, should be made such that 4 to 6 different genets are planted close to each other. That then allows for sexual reproduction, fertilization between eggs and sperm during broadcast spawning events. These recommendations were the result of ongoing discussions with the Coral Genetic Management Group that I chair for the Coral Restoration Consortium, and this has really been a wonderful group of people to meet with. So I'll maybe mention very briefly what role the coral symbiotic algae play in coral adaptation, because people usually are curious about this and ask. So let me give you just a really brief overview how or what the role of the coral algae may play in this. So zooxanthellae, or algae in the family Symbiodiniaceae, make up a significant portion of the holobiont biomass, and this is data from Kemp et al. And they literally weighed the Symbiodinium cells in the coral tissue. And in the summer, the percent of the holobiont biomass can be significant, between 10 and 20%. In the winter, it's a little bit lower. Now, not only do these algae make up a significant amount of the holobionts biomass, they are usually of just one species of symbiont associated with the coral host. There are differences. Some coral hosts can host more than one species of symbiont, and sometimes environmental conditions can shift this symbiosis by allowing other species to invade the coral tissue. Here, this is an example of the staghorn coral along the Florida Reef Tract, and offshore fore reef and mid-channel reefs, usually the colonies hosted Symbiodinium phytae, but in inshore turbid reefs, the diversity was higher. There was also algae of the Cladocopium, genus Cladocopium, as well as Durstinium trenchi. Now, this change is often temporary, but especially this association with Durstinium trenchi is really interesting. So Durstinium trenchi is thought to be an invasive species, algal species in the Caribbean. And here we used PAM fluorometry to measure coral health in response to heat stress trial again. This work is led by Richard Karp at the University of Miami. And so Rich had two groups of Acropora palmata colonies. One group hosted the homologous symbiont Symbiondinium fiti that we usually find in this coral, and another group hosted the invasive symbiont trenchi. And after a 40-day heat stress, the colonies hosting fiti had a 70% greater decline in photochemical efficiency for the same amount of heat stress than the colonies hosting D. Now, the question is always, is there, these are encouraging results, obviously, because it increases the heat tolerance of the holobiont, but are there trade-offs? For example, slower growth rates when these corals associate with the invasive symbiont. These here are laboratory results, and indeed, initially, the corals hosting Durstinium in the orange bars had lower growth rates than the ones hosting the native fiti. But over time, this disadvantage decreased. So we went from a big advantage for S fiti at the first time point to a reversal where the trenchi hosting corals sort of caught up or even exceeded the growth rates of the ones hosting fiti. This is indeed great news, but the question is still, how does this translate into wild populations where we usually find that the corals will switch back to S fiti over time? This could very well be because environmental conditions haven't yet changed to a point where hosting Durstinium is really advantageous over the whole year. So to summarize, how do corals adapt? Well, we see that selection is shaping coral populations as we speak. Selection acts on the phenotype of coral genets, such as heat stress, as well as disease resistance, which I haven't shown you, but there's publications out there. Adaptation can happen to a population when coral genets with a selected phenotype pass that phenotype on to their offspring. We can see that selection causes shifts in allele frequencies in coral populations. The environmental conditions that we're looking at right now are causing shifts in the associations between coral genets and the symbiotic algae, and this is resulting in new holobiont phenotypes, and how this plays out over time, we have yet to fully appreciate. So I hope I've shown you in this presentation how holobiont-standing genetic diversity fuels adaptation on reef-building corals. And while we have a general understanding of how adaptation may happen, much work remains to be done to refine these results. And the terrestrial literature also shows us that holobiont diversity fuels diversity in associated species. So, for example, in associated invertebrates, or even fish. Some forays have been made to study these interactions in corals, but there's still a lot of room for additional discoveries. So these advances in understanding of coral adaptation underscore the need for broadly available standardized methods to find stress-resistant corals, to conserve genetic diversity, and to use these resources to guide genetic management plans. Let me close by saying that coral adaptation is ongoing, but without immediate and decisive action to curb carbon emissions, our coral reef ecosystem in their current form will not survive the next decades. And we have a moral duty to stop carbon pollution. It's critical for our own survival. Having said that, the commitment of scientists and managers and practitioners that we are seeing currently has already yielded great progress towards safeguarding the genetic variation in corals and to maximize their chances for adaptation to a changing world. Thank you.
Thank you very much, Iliana. Virtual round of applause from everyone, I'm sure. Yeah, thanks very much for this, for your talk, for the broad introduction. That was really good, to have a bit of a refresher there as well for everyone who might not be so familiar with these concepts, and for your closing words as well. I think it's really important and we can't say it often enough. I'm wondering whether there are any questions, but I'll hand over the moderation of those to Jonathan because for some reason I can't see the chat. So if you have any questions, raise your hand or write in the chat, please.
Yeah, great, thank you. We haven't got any questions yet in the chat, but I see Alice has put her hand up, so
Yeah.
Alice, over to you.
Thanks, Jonathan. It's a really, really interesting talk. Thank you ever so much for that. I'm involved in a project out in Bali where we're doing a lot of artificial reef coral restoration. So we're not doing active transplantation. We're just kind of putting structure into an area that has been degraded. So it's a sand flat that we're building structures onto. And one thing I was interested in, is there any kind of like priority effects in terms of the species that we might potentially colonize, and with the ones that are more heat tolerant or larger growing, do they tend to have like a quicker reproductive strategy or anything like that?
Yeah, I think it kind of depends on where you're at exactly in the world. We do know that there's certain species that's, without heat stress, there's certainly a succession, in a coral reef community, and these are sort of traditional, studies that have been done since the early 70s or so, and under heat stress, you would, depending on how frequent your heat stress is in the recovery period, you probably get other species recruiting to your reef, certainly. So, but it also really depends on what's around, right? If there are no local sources for coral larvae, then you're reliant on long distance dispersal, and so that just really depends on the particular situation.
Yeah, luckily we are right next to a natural coral reef, so we have got that active larval supply, and it's incredible how quickly it has colonized and how quickly the coral is developing. I mean, at the minute we haven't had, so like over the past couple of years, we haven't had big bleaching events like I know that have been happening around the world. But I'm just thinking long term, if we can try and encourage that resistance, it would be beneficial.
I think so. People are talking about when we're starting to think about restoring really denuded areas. There's been a tendency towards trying to encourage settlement of not just the corals themselves, but the other components of the ecosystem as well. So sometimes people will put in these settlement plates, basically in healthy reef habitat. And they will get colonized with all kinds of things, not just coral larvae, but with certain microbes and invertebrates, and then use that to move them to your denuded area to sort of try to jumpstart the process of getting sort of a more complete ecosystem established. That kind of, again, depends on how far away your reef is and how necessary that really is, but in some cases this can apparently really support recovery.
Well, thank you very much.
Great. Well, we don't have any other questions. Maybe I'll ask a question if that's all right. This is obviously not my field, so it might be a bit of a naive comment, but I was wondering about the applicability of your work to cold water corals, and we have a lot of cold water corals around the coast of the UK and Europe as well. So, is there any sort of applicability to those systems?
Yeah, cold water. I've done a bit of work with cold water corals in the Gulf of Mexico that were impacted by the Deepwater Horizon oil spill. You might remember that being a quite huge event. And cold water corals are characterized by even slower growth rates than the shallow water corals, and we have even less of an idea about how they reproduce and recruit. And this is certainly challenging in that regard. Now, the principles, however, should apply, right? We know that deep water corals often also reproduce asexually, quite extensively, and having whole fields of genetically identical, these fields of clone mates, that certainly would prevent or is a barrier to restoration. If you don't know that your coral that you're trying to restore is extremely clonal, then your collections may include, just a few genets. And we know from so many fields that restoring with only a few genets is detrimental, right? They'll have less disease resistance. They'll, in general, be less resilient. So in that case, I think this work does, like the shallow water work and the bit of deep water work we've done, certainly says that you ought to develop some tools to survey your genetic diversity before you do anything else. Now, it's just the work, the time it takes to do any kind of manipulation in deep water is just so much longer. So, but people are doing it. I mean, people are working on trying to get better, like tools, ROVs, and so on, to help us even restore these deep water coral assemblages. And they certainly are impacted by not just oil spills, but all kinds of other events that we might miss because we don't look as much, right? We don't have eyes on these ecosystems the same way we do on the shallow waters.
Excellent, thank you. I think I can see that Michelle is typing in the chat, but maybe while that's happening, Steffi, it looks like you've got a question.
Yeah, I have another question. Again, this is not my field. We're testing how salt marsh plants are affected by higher temperatures, and we see that there's often an interaction of multiple factors like elevation, elevated CO2 and temperature, or drought and temperature. Is there anything going on where you think additional climate change factors and how they interact with warming would be something to look into?
Yeah, it's a good question. I mean, there was a lot of worry about ocean acidification for corals for a while. The impacts of ocean acidification on coral survival are not as clear cut, I guess. So you can't universally say that decreasing or increasing ocean acidification is going to have this or that effect on all these corals. So the effects are quite nuanced, I suppose, and variable. However, I do worry about interaction effects. So maybe not ocean acidification so much, but definitely emerging diseases. And we've had a few of these. For example, stony coral tissue loss disease has devastated certain coral species in the Caribbean lately. Now, interestingly, there's some indications that it could be that the disease is actually the symbiont that is really affected. So nevertheless, though, these emerging threats go beyond just heat stress. And that is, I think, where it's really important to not just select corals because of their temperature resistance, but we need to keep we need to maintain genetic diversity against these emerging threats. And I think this is a lesson learned in terrestrial and even in the animal breeding realm for certain. So it's having this mixed portfolio. I think that's our best bet in that case.
Great. So we've got a couple of more comments in the chat. So Sally said, thank you very much for the talk. And she's going to get all of her final year students to watch this. And Michelle says, great talk. Thank you. It seems that is all of our questions for today. Okay. Yeah, thank you once again for the really fascinating talk. Our next webinar will be given by Anna Stoic from the University of Essex on comparing alternative methods to estimate the importance of estuarine nursery areas to commercial fishing. So hopefully everyone can join us then, and we look forward to that talk in April. So thank you, everyone.
Thank you. Bye.
Thanks. Bye.
Thank you. Bye.